Good morning. It's February 5th, and this is your daily brief in science. Here's everything you need to know. A groundbreaking study from the University of Central Florida is shedding light on the elusive early years of sea turtles, a period often referred to as the lost years. This research, led by Kate Mansfield, has utilized satellite trackers to investigate young sea turtles during a critical life stage that spans from 1 to 10 years after they leave the beach. Published on February 3, 2025, the study involved tagging 114 young turtles over the course of a decade. The research focused on endangered species, including green turtles, loggerheads, hawksbills, and Kemp's ridleys. To gather meaningful data, the team developed innovative flexible solar-powered tags capable of remaining attached long enough to transmit location data before detaching due to the turtle's rapid growth. The GPS tags provided location data for varying durations, revealing significant insights into the turtle's behavior. Contrary to previous beliefs that young turtles passively drift with ocean currents, the study discovered that they actively swim and make decisions regarding their movements. Co-author Nathan Putman confirmed this active behavior by comparing the turtles' locations with drifting buoys, noting that while many buoys washed ashore, the turtles did not. The findings, published in the Proceedings of the Royal Society B, highlight the variability in the turtles' locations as they transition between continental shelf waters and the open ocean. This research enhances biologists' understanding of the Gulf of Mexico's critical role as a habitat for several endangered sea turtle species. It emphasizes that scientists had lost track of the turtles, not that the turtles were ever lost. Understanding how young turtles utilize the Gulf of Mexico is crucial for their ecology and conservation, particularly given its importance for 4 endangered species. Overall, this research significantly contributes to our knowledge of sea turtle behavior and the ecological dynamics of their habitats. On February 4, 2025, planetary geologist David Kring from the Lunar and Planetary Institute unveiled groundbreaking findings about the formation of lunar canyons. The study, published in Nature Communications, utilized data from NASA's Lunar Reconnaissance Orbiter and advanced computer modeling to analyze a significant impact event. Research indicates that a 3.8 billion-year-old impact in the Schrödinger impact basin was responsible for creating these canyons by ejecting rocky debris into space. The object that caused this impact was approximately 15 miles in diameter, much larger than the asteroid believed to have led to the extinction of the dinosaurs. The energy released by this event was immense, estimated to be about 130 times greater than the current global stockpile of nuclear weapons. Unlike the Grand Canyon, which was shaped by water erosion over millions of years, these lunar canyons were formed in roughly 10 minutes from debris generated by the impact. The canyons, named Valles Schrödinger and Valles Planck, extend 270 and 280 kilometers in length, reaching depths of 2.7 and 3.5 kilometers respectively, surpassing the Grand Canyon's depth. The debris from the impact traveled at astonishing speeds, estimated between 2,125 to 2,860 miles per hour, significantly faster than a bullet. The canyons converged toward the southern edge of the Schrödinger basin, indicating that the impacting object approached at an angle, splashing material primarily northward. This region contains some of the Moon's oldest rocks, potentially dating back to its formation, which could offer valuable insights into the Moon's geological history. These findings hold considerable importance for future lunar exploration, particularly for NASA's Artemis mission, which aims to land astronauts near the Schrödinger basin. The uneven scattering of debris suggests that astronauts landing near the South Pole Aitken Basin will have an easier time collecting samples from the Moon's early geological history. A recent study published in Nature Medicine has uncovered alarming changes in biomarkers among individuals who have had COVID-19. The study found a lower AB42 to AB40 ratio, increased p-tau 181, and elevated neurofilament light chain, or NFL, levels. These biomarkers are indicative of Alzheimer's pathology and suggest a potential link between SARS-CoV-2 infection and an increased risk of Alzheimer's disease, particularly in older adults with pre-existing health conditions like hypertension. Professor Paul Matthews highlighted the long-held suspicion that infectious diseases may contribute to the progression of neurodegenerative diseases. He emphasized the need for further research to clarify causal relationships and identify modifiable risk factors for dementia. The authors of the study also stressed the necessity for additional research to validate their findings and to explore preventive strategies for at-risk patients to mitigate post-COVID neurological risks. The researchers acknowledged limitations, including insufficient data on the severity of COVID-19 infections and the novelty of the blood biomarkers for amyloid and tau, which are still being evaluated for clinical relevance. To enhance the study's accuracy, participants were matched based on age, sex, ethnicity, and location, which helped minimize confounding factors. Dr. Eugene Duff, the study's first author, noted that inflammation resulting from COVID-19 might drive changes associated with neurodegenerative diseases, although the exact mechanisms remain unclear. The study observed altered inflammatory markers in post-COVID individuals, suggesting a prolonged inflammatory response that could contribute to brain pathology. Notably, changes in biomarkers were detected even in individuals who experienced mild or asymptomatic COVID-19, indicating that the effects on brain health are not limited to severe cases. Previous research has indicated that systemic viral infections could heighten dementia risks, which reinforces the findings of this study. However, it's important to note that these findings are based on observational data and do not confirm a causal link between COVID-19 and dementia. This raises further questions about whether similar effects might arise from other infections, such as influenza or pneumonia. Researchers at Houston Methodist have made significant strides in the fight against metaplastic breast cancer, or MPBC, a rare and aggressive form of the disease. A national study published in Nature Communications has identified 2 inhibitor drugs that may effectively treat this challenging cancer type. The study reveals that a combination therapy using a phosphoinositide 3 kinase inhibitor, or P13K, alongside a nitric oxide synthase inhibitor, or NOS, can disrupt 2 distinct signaling pathways involved in MPBC. The findings highlight that MPBC tumors exhibit heightened co-activation of the PI3K and NOS pathways, which contributes to their aggressive behavior. This dual targeting approach is proposed as a more effective treatment strategy, especially since MPBC often resists conventional therapies. Dr. Tejaswini Reddy, the first author of the study, expressed hope that these insights will lead to specific care plans for metaplastic cancer patients and improve their long-term survival rates. Dr. Jenny Chang, the corresponding author, emphasized the importance of these findings for enhancing treatment options for patients who currently have limited choices. Treatment for MPBC typically mirrors that of triple-negative breast cancer, but the responses in metaplastic cases have been poor. The aggressive nature of MPBC, characterized by rapid growth, frequent spread, and recurrence, complicates effective treatment. While the researchers acknowledged the small sample size of tumor biopsies, they stressed the necessity for further studies to validate their findings. A Phase 2 clinical trial funded by the National Cancer Institute is set to investigate the therapeutic combination discovered in this preclinical study. Tumor biopsies from patients who responded to treatment showed increased tumor differentiation and decreased cancer stem cell markers, indicating the potential success of this combination therapy in targeting resilient cancer cells. Researchers at UC San Francisco have made a significant breakthrough in cancer treatment by using CRISPR gene editing technology to convert white fat cells into calorie-burning beige fat cells. This innovative method, led by Nadav Ahituv and postdoc Hai Nguyen, focuses on activating dormant genes within white fat cells to create engineered beige fat cells that aggressively consume nutrients. These cells are specifically designed to target nutrients that certain cancers preferentially use, suggesting a tailored approach for treating different cancer types. Laboratory experiments have shown that this method is effective against 5 types of cancer, including breast, pancreatic, and prostate cancers. In vivo studies with fat organoids indicated that the implanted beige fat cells were able to suppress tumor growth in mice that were genetically predisposed to cancer. Collaborating with breast cancer specialist Jennifer Rosenbluth, The researchers tested the engineered fat cells using patient-derived tissues, achieving promising results by outcompeting breast cancer cells both in vitro and in vivo. The engineered beige fat cells significantly diminished the survival of various cancer cells, including those from breast, colon, pancreatic, and prostate cancers. Notably, these fat cells were effective even when positioned far from tumor sites in mice, as they aggressively consumed nutrients, depriving tumors of essential fuel for growth. Published on February 4, 2025, in Nature Biotechnology, this research highlights the advantages of fat cells for cell therapy, such as ease of extraction and favorable behavior in the body. The potential applications of these engineered fat cells extend beyond cancer treatment, with implications for diabetes management and iron regulation in conditions like hemochromatosis. This pioneering approach draws inspiration from established medical procedures like liposuction and plastic surgery, which may facilitate quicker clinical application as a cancer therapy. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.